Okay, so this is the final topic. This is on mutations, changes in the genome. And we're gonna focus on B. I'll just really briefly mention A, but I would actually like you to take a look at what A is. A are examples of what are called chromosomal mutations. So I want you to take a look at that. Next time we'll meet, we'll discuss them, okay? But these are mutations where, for example, this is something called a karyotype. This is a normal karyotype. This is an abnormal karyotype. So one type of mutation is when you get either an extra copy of a chromosome. You could see that case, there's an extra copy. This is called trisomy 21. So trisomy 21 means, tri means 3. So the 21st pair of chromosomes actually has an extra one. So it's got 3. This is actually what leads to Down syndrome, okay? It's not the only scenario. I mean, you can have trisomy event, really any chromosome, but that one is associated with Down syndrome. It's a result of when chromosomes divide during cell division. They're supposed to divide evenly, but sometimes they don't, and you can see here, oops, that has not happened. Whereas this cell gets an extra copy, and this cell actually is missing a copy. So chromosomal mutations are when they don't. One type of chromosomal mutation is when they don't divide properly. Here's just a list of certain types, which you really don't have to know them. Okay, so I'm going to get you to look at the rest of the examples of chromosomal mutations. So I'm going to skip over this, okay, because I want to actually focus on mutations in the actual DNA sequence. So this, in other words, assumes that the number of chromosomes have divided properly, okay, and these are mistakes in the DNA sequence itself. So not in the number of chromosomes, but in the actual DNA sequence. And we're going to look at a few examples of this. Okay, we're going to look at two types. We're going to look at mutations that exist in the coding section. So we can, if you remember, the coding section is the part that gets translated eventually into protein. Okay. So first we'll talk about mutations in the coding section. All right, and they could be classified as either silent, missense mutations, nonsense, and something called frame shifting. Okay, so let's take a look at what this is. So imagine that the blue is your normal DNA sequence, right? Now we know how transcription works. We take the template strand, you read it 3 to 5 prime, right? And then you build RNA 5 to 3 through complementary base pairing, right? So if you read TAC, you put in AUG, that's transcription, right? And then we know that you take your transcript, and if it's eukaryotes, we modify it a little bit. We do a little bit of cutting, capping, and tailing, but eventually it gets translated into a protein, which is what you're seeing right here. So AUG becomes methionine, ACU becomes threonine, and so on. And if you recall, this is because there is a genetic code. Let me pull this up. Actually, I can probably, from this one, is actually a nice one. Let me pull the table from here. If you remember, there is a genetic code that is used to translate the codons into an amino acid, right? So AUG is methionine, AUU is isoleucine, I think it's isoleucine. CUU is leucine, and so on and so forth. Okay, and you see some stop codons. Now, so this is what normally happens, okay? Transcription and then translation. Now, if you have what's called a silent mutation, then, so a silent mutation says that you get the same amino acid, okay? So what that means is, imagine that we had, let's say we look at ACU, okay? So here is my genetic code. So it says the ACU gives you threonine. So if I look at ACU, you can see the ACU gives you threonine. Now, in a silent mutation, if the last letter, okay, let's say instead of it being ACU, the last letter, the U, gets changed to a G, okay? Now, if that happens, then you still get threonine. You get the exact same amino acid. So if I change ACU to ACG, I still get threonine. Nothing happens. Okay? So actually, I don't have a slide of that. I thought I did have a slide of that. I don't. Okay. So in a missense mutation, if you change a letter and you get the same amino acid, that's considered a silent mutation. Now notice that it's often the third letter. Like for example, if I look at the code for serine, you'll notice that as long as it starts with U and C, the third letter actually is irrelevant. So if I have a sequence, an mRNA sequence, that is supposed to be UCU, but the third letter changes to UCC, I still get serine. So that's called a silent mutation, okay? And that's often the case with the third letter in the codon, all right? By the way, these are what are called point mutations. I should mention that. So silent mutations and missense and nonsense, these are generally what are called point mutations because they happen at a very, very specific point, okay? So that's silent. A missense mutation is when you get a different amino acid. So for example, this, you can see the third codon is supposed to be AGA. Now, if a mistake is made and it's now ACA, so the letter G is a point mutation, okay, a specific point in the DNA sequence, where the G now is a C for whatever reason, because mistakes do happen in this process. It's rare, but they do happen. Then that gives you a new amino acid. ACA, you can see here, where is it? ACA is threonine, all right? The previous letter, you can see give you a different amino acid. So we went from arginine to threonine. That's called a missense mutation, all right? And if you recall, this actually is what happens in sickle cell anemia, right? Was one mistake, changed the amino acid. But if you remember, we talked about proteins, sometimes if you change the right amino acid, you affect a very important bond. And if you affect that bond, you could change the shape of the protein, and it could be a very, very, very significant type of mutation. Here's another example. You can see the 12th codon in a protein called RAS, all right? The protein's codon, the codon that gives you glycine is GGC, but there's a mutation that gives you GTC. You can see it's the second letter, because if I change the third letter, most likely it doesn't make a difference. And but in this case, change the second letter or the first letter, you're probably going to get a new amino acid. So instead of glycine, now you get valine. What that does is that actually affects this RAS protein that you're seeing here. Okay, this is the RAS protein. Now, the way the RAS protein works is it has a domain. So this is something that's called GTP. GTP is a cousin of ATP. Now, the RAS protein, what it's supposed to do is supposed to take the GTP and break it down into GDP to turn itself off. So right now, this is considered the on state of the protein. So this protein right now is on, which means it's working. When it's working, what it does is it actually activates transcription factors. Now, if you remember, transcription factors are proteins, regulatory proteins that control genes. In this case, these transcription factors turn on the genes for cell division. So what happens then is this cell undergoes mitosis, starts dividing. Now, you learned back in grade 10 that cells want to divide under certain conditions. When they're growing or when they're damaged, they want to repair themselves, they divide. But this process has to be controlled. It has to be able to turn itself off. So the way it turns itself off is the RAS protein has a domain that binds to the GTP and cuts off one of the phosphate and becomes GDP. And when it does that, it actually switches off. That mutation, though, that very specific mutation, affects the ability of RAS to do this, which means that RAS stays on. If it cannot convert GTP to GDP, it stays on, which means then it's always producing active transcription factors to promote cell division, which then means that this could lead to what kind of disease? What do you think, ladies? This protein basically activates transcription factors for cell division. And if it cannot turn itself off, that means it cannot turn off cell division. So what kind of disease then could this lead to?
Cancer.
Yeah.
Cancer?
Yep. Yeah. Very good. Very good. Very good. Excellent. So that's another example of a missense mutation, okay? The third type is a nonsense mutation. Now, what happens then is instead of a mistake in the first or second letter giving you a new amino acid, the mistake actually gives you a stop codon. You can see that's supposed to be UGG, right? But UGG, if I change that second letter, you can see UGG is right here. Sorry. If I change UGG, if I change that second letter to, sorry, is it the second? Yeah, it was the second letter was changed in the example, right? In this example, the second letter, yeah, second letter is changed to an A. Okay. So if I change that letter to an A, you can see that it actually produces what's called a stop codon. Interestingly enough here, if I do change that third letter here, it can also lead to a stop codon or another amino acid. So this is actually one of those few examples where if you change the third letter, you actually produce a new amino acid. Okay, but what happens in that case is you get a stop codon. So the protein ends very premature, and this is called a nonsense mutation. Now this is actually if you remember, mutations sometimes, you know, they don't make a difference because they, like even a mutation that gives you a new amino acid might still not make a difference to the protein. Do you ever remember why it might not make a difference to the protein? What was the reason behind that? Well, yeah, I mean, so if it's a silent mutation where you get the exact same amino acid and the sequence doesn't change, obviously that cannot affect the protein, right? If I change the third letter, most likely nothing happens. I get the same amino acid, therefore the protein will be the same. If I get a missense mutation, we talked about, you know, like sickle cell as an example, we just talked about RAS. But the thing about a missense mutation is even if I get a new amino acid, I might still not see any effect on the protein. Do you remember what the reason behind that was? Dorcas, what was the reason behind that? Remember?
Some amino acids are like the same type.
Yeah, they might have,
or
they might have the exact same type of R group, right? So like, some R groups are negative. Maybe the new amino acid also has a negative R group and therefore does not affect any bond it's involved in. Uh, maybe the amino acid isn't very important. Maybe it's like the color of a key. You change the color of a key. Does it really matter? No. So missense mutations can still behave like silent mutations, even though you do get a new amino acid. But if I look at a nonsense mutation, that do you think it's likely that a nonsense mutation will be silent? Like, in other words, will have no effect. I mean, you are missing a chunk of the protein. Do you think it's more likely that that's going to have an effect, or do you think it's less likely that it's going to have an effect? Julie, what do you think?
So you asked if it had more effect, like
Yeah,
more.
we just talked about a missense mutation might not have any effect at all, right? But a nonsense mutation where you're missing a chunk of the protein,
So it will.
yeah, I would say it's gonna, have a major, because you're missing, chunk of the protein. The odds are it's gonna have some sort of an effect. Okay. The
Yeah.
The fourth type of mutation, if I go back to mistakes within a DNA sequence, is something called frame shifting. Okay. Now, and by the way, if if you're doing your lab on the virus, the viral genome, you probably would have seen lots of examples of these point mutations where letters have been changed. Now, I didn't ask you to tell me if they're silent or missense or nonsense, right? That was not part of the lab, right? But you obviously saw point mutations, right? When you compare one virus to another virus, you notice that some of the letters get changed, right? So some of those letters will, they'll be silent mutations, some will be missense, none of them probably will be nonsense, because that would really be bad for the virus. So you're most likely going to see silent and missense mutations when you get those substitutions. But the next one, you may have seen something that looks like what I'm going to show you, something called frame shifting, okay? And this happens when you get either an insertion or a deletion, all right, of a letter, or maybe two letters. It doesn't have to be one. It could be two, it could be three, it could be four, it could be five. And I'm going to show you. So here's an actual slide of what it looks like, okay? So the example here is, the top example is two letters gets removed, and the one below it is one letter gets removed, and it could lead to a whole host of problems. And I'll show you why. I'm going to show you why with an example using a statement, okay? So I'm going to write something on my canvas. Here we go. A big red fat cat ate the red bat. Okay? I think all of you can you see that? Is it good? Frankie, can you see
Yes.
that? Yeah?
Yes, sir.
All right. Okay. Now, oh, sorry. I gotta change something. I can't have the word A. I'll tell you why. Because we're gonna read this three letters at a time, okay? So I actually have to change something. Hold on. Let me erase that. It's supposed to be the big red fat cat ate the red bat. Okay. Now, the reason why I wrote this is because if you remember, the mRNA, when it's translated three letters at a time, because the three letters are a codon, right? Each codon gives you one amino acid. All right. So that's why I wrote this as three letters. That's why I had to take out the letter A, or the word A. Okay. Now, if I make, for example, let's say we talked about like a silent mutation. So I'm going to show you an example of a silent mutation. Although this is not the best example, but if I change the C to a K, and I say this sentence, the big red fat cat ate the red bat. Did it sound any different if I changed the C to a K? No. So that would be like a silent mutation, right? This is an analogy. The problem is, as you saw with the genetic code, if you change the first letter, it's not going to be silent, right? First letter is very, very important. So, but this is just an example to help you understand the difference between frame shifting versus the other types of mutations, okay? If I, for example, change the sentence, so if I wrote like this, if I wrote the big dead fat cat ate the red bat, then you could see now it's not silent. It's not a silent mutation anymore. This is a missense, right? Because now the codon red has changed to dead, right? So it sounds different, right? That would be an example like of a missense mutation. Now, in frame shifting, what happens is you add a letter or 2 or 3 or 4, or maybe you subtract a letter. So watch what happens when we do this. Now imagine that the original statement, okay, go back to the original statement, which was this, right? The big red fat cat ate the red bat. But imagine now a deletion happens. So a letter gets deleted, gets cut out, okay? Now, when I go ahead and read this, look what happens. Remember that the ribosomes read 3 letters at a time, right? So the, big, edv, atk, ata, tet, her, ebd, at. What happened to the rest of this sentence? All messed up. And it's the same problem if instead of taking away a letter, what if I add a letter? Well, look, it's the same problem. The, big, r, dva, tisa, I don't know how to say that, tat, eth, dva, same problem. So what frame shifting does is it not only changes, or most likely changes the amino acid at that particular point, but it changes everything after that. That's why it's called frame shifting, because the actual reading frames get shifted depending on if you're inserting or deleting. Now, do you think this is going to be A, have no effect, or B, have an effect? What do you think? Yeah, this is definitely going to have an effect, right? So frame shiftings are like insertions and deletions are really, really bad, except in the non-coding section. Now, when you look at the viral genome, did you notice that some viruses had insertions and deletions that looked like chunks were missing? There are some samples that I've asked you to look at, where when you look at them, the virus looks like it's missing material, okay? Looks like, something's either gone missing, it's either been deleted or it's been inserted, right? Now, this happens though in the non-coding section. Now, why the non-coding section? Because if you remember, if I go back to the structure of a gene, okay? Let's go back to the structure of a gene. So here's my gene, right? The coding part is the part that gives you a protein. And that's from, if you remember, that is from start up here all the way to stop, right? So all of that is a protein. Now, all of this is read 3 letters at a time. So if I was to get imagine an insertion right here. Okay, imagine the letter G was inserted right there. So your first amino acid, ATG, is fine. But now everything after that point, everything is affected. Everything. So now the next codon would be, it was GCC, but now it's GGC. And the next codon was supposed to be GGC, but now it's CGG. So everything gets affected. But if you have insertions and deletions in the non-coding section, it's actually okay. It might not make any difference at all. If I take away a letter here, or maybe I take away 2 letters, or maybe I add some letters here, or in this region, this does not get read 3 letters at a time, which means insertions and deletions in the non-coding areas are tolerable. They're probably not going to have any effect whatsoever. Okay, that's most likely true, but we can talk about next where that might not necessarily be true. Do you have any questions at this point about these 4 types of mutations? Again, this is in the coding section. So we're talking about mutations in the part of the gene that gives you the protein, the part that gets read 3 letters at a time. Okay, and the 4 mutations we just covered are silent, missense, nonsense, and frame shifting. Any questions about those mutations? No? Okay, so then if there's no questions, then what we can do is talk about what happens then about, like, what about mutations in non-coding sections? All right, that's what actually we're going to talk about next, which we actually covered a little bit before. And if you remember, the mutations in non-coding sections, those are the types of mutations that would affect, if I go back to, for instance, we talked about gene regulation, okay? The coding section would be here. Oops, sorry. The coding section would be the part that gives you the protein. So that would be here, right? This section. The non-coding section is the part that controls transcription. So this is the part involved in regulation. So mistakes here, if you remember from last time, what they do is they don't affect the protein, but they can affect the regulation of the protein, whether or not the protein gets turned on or gets turned off, or how much, right? And we talked about an example of that was lactose tolerance, where there's, in that case, the lactose tolerant individuals are actually the mutants, and there's a mistake somewhere in the non-coding section that leaves the gene on, where it's supposed to normally turn off at about the age of, I think, about 5 or something like that. So let's talk then about the mutations that are in the non-coding section, and what that, like, how that's important, okay? Because here's the thing about mutation. What would happen if a mutation knocked out the promoter region? What do you think would happen if a mutation wiped out the promoter region? What do you think would happen, girls? What's the role of the promoter region? Adele, do you remember? Is Adele still there? Adele, you there?
Yes, sir.
Do you remember what the promoter region does?
No.
No?
Is it the transcription?
Yeah, it controls the turning on of the genes, right? Because RNA polymerase reads it and then converts it into protein eventually, right? Because you get transcription and then that becomes translated into protein. Now, what happens if the promoter region is deleted? So if I go back to this picture, right, and I say, imagine a deletion, some sort of a mutation that cancels out the, that cancels out the promoter region, just wipes it out. So it's gone, disappears. All right. Now, what happens in that case? What would happen to gene Z, Y, and A? Would they ever get switched on? Considering that the promoter region has to be recognized by RNA polymerase, if the promoter region is gone, then what happens? The gene stays off. So mistakes in the non-coding section, again, are going to affect whether or not the genes are being used. So what happens then if you get mistakes in the non-coding section that, let's imagine that mistake kills the promoter region, what that could lead to is something called a dead gene or what's called a pseudogene. So a pseudogene is a dead gene that could be caused because that non-coding section acquired some really devastating mutation. For example, the mutation destroys the promoter region, okay? And what happens then is that gene eventually becomes something called a pseudogene, and it just starts to accumulate lots and lots more mistakes. So here's a slide that's showing you a gene. It's called RPL21. Actually, a gene involved in making ribosomes. Ribosomes are part protein, part RNA. That little orange copy is the only working copy of this gene. All the other blue bands are locations of the pseudogenes, the dead genes. Now, there are many ways to kill a gene. I just mentioned one, if the promoter gets wiped out, but any really devastating mutation in even the coding section, like any really big deletion or insertion, will also kill a gene. So there's many ways to kill a gene, right? And if a gene gets killed, it's called a pseudogene. Here's an example of one. You might remember this from last year. We talked about this is the gene that is responsible for making vitamin C. Now in genetics, a dead gene, a pseudogene, has a symbol, okay? That symbol is right here. It's a little trident symbol. Just looking for my pen. I can't find my pen anymore. It's disappeared on me. Gone. Okay. Do you see that little symbol here? Sorry. So right here, this little trident symbol. Sorry, now my pen just disappeared on me. That's the symbol for a dead gene. So when you come across that in genetics, that means the gene is no longer with us. It's resting in heaven. Now, in humans, gulo is a pseudogene, but not in mice. So what gulo does is gulo actually makes the genes that make vitamin C. So it actually encodes for an enzyme that can convert glucose into vitamin C. And in primates, this gene is gone. It just died. Okay. Now, the reason why a gene dies is very simple, actually. The mechanism is, there could be many reasons, many causes for death, right? The mechanism could be loss of promoter, maybe some really devastating insertion and deletion. But the reason why it happens is the same. And it happens because the gene is no longer useful. Okay? You can think of it as use it or lose it. This is the idea. So the reason why it's allowed to happen is because the gene isn't really important anymore. Now, in primates, the reason why the gene wasn't important is because primates were getting lots of vitamin C from their diet. So a dead gene for making vitamin C would have no difference to primates, would make no difference to them because their diet is already rich in vitamin C. If a gene is useful, though, then it will never become a pseudogene. So you can imagine the gene that builds hearts, okay? It's got the unofficial name of Tin Man. Now, if you remember Wizard of Oz, Tin Man wanted a heart. You remember the story Wizard of Oz? Tin Man wanted a heart. Okay, and I can't actually remember the actual name for this gene, but it goes by the name Tin Man also. Now, what would happen if Tin Man died? Like if that gene died in an organism, then what would happen to the organism? Well, the organism would not survive, right? So if Tin Man dies, let's imagine some mistake, right? Whether the promoter region of Tin Man gets wiped out or some massive deletion in the coding section causes Tin Man to be a dead gene, okay? A pseudogene. Then what happens is that organism will not survive. The organism dies. And if the organism dies, that dead gene doesn't get passed on to the next generation. It's not going to happen. It's not possible. That's not going to happen. So the only way a gene gets passed on to the next generation is if the organism survives and passes on copies of its DNA. So if a gene is a dead gene and it's getting passed on, because gene's no longer important. So let's look at some examples. Some other examples. So here are two examples. So we're going to look at color. So often fish or organisms that live in caves, after many, many, many years, I don't know how many years, not like you put a fish in a cave and the next day it loses color, but after many, many generations, organisms that have adapted to caves tend to be albinos or have no color. All right. Now, why? What's going on here? Well, is color important in the dark? What do you think? No, it's not important in the dark. So imagine a gene. Okay, we'll call it gene A. Gene A gives color. Now, gene A dies, becomes a pseudogene. Maybe its promoter region gets destroyed. Now that fish has no color. No color. Now, does it matter? The answer is no. It's not useful. Color is not useful. So can that pseudogene get passed on? Yes. It's the same story for eyes. You notice these cavefish have no eyes because what's the point of eyes in the dark? So imagine gene A didn't code for color, it coded for eyes. Now, gene A dies, no eyes. Does it matter? No, it's not useful. So therefore, if it doesn't matter and it's not useful, then the gene could die and it can be passed on to the next generation. I mean, color is only going to matter if you can see it. If you can't see it, color ain't going to matter. Okay? Now, these are examples of, losing. I'll show you one more. These are examples of losing genes. I'll show you one more example, and that is with seeing color. Seeing color. It's not just color, not just eyes, but actually seeing color. Okay, so we have 3 genes that allow us to see 3 colors in particular: red, green, and blue. Okay, red, green, and blue. Now, most mammals have only 2. They can see blue and they can see yellow very well. So like a dog can't see color as well as we can because they have 1 less copy of the gene that codes for color, or sorry, codes for seeing color. Okay. Now, what's interesting is if you look at things like lamprey, which are like they evolved a long, long time ago, they have 5 genes for seeing color. So they see color really well. Birds, reptiles, and fish have 4. Now, mammals, for some reason, have 2. But we know mammals evolved from things that look like reptiles. So they used to have 4, but they lost 2. So in other words, those 2 genes became pseudogenes. They died. Okay, so why were they allowed to die? Obviously, this must be the same story as the fish, right? Obviously, in this case, color was not as important. So some of those genes, 2 of them, died. Why weren't they important? Because mammals evolved during the time of the dinosaurs. Now, mammals were big. Sorry. Dinosaurs were big. Dinosaurs are scary. Mammals are tiny, like literally smaller than mice, maybe mice size. What do you think the mice were hiding when the dinosaurs were roaming this planet? Where do you think they were? They were under?
Ground.
Yeah, very good. They were underground. They were below ground. Dinosaurs were above ground. They were below ground. Not only that, but they were also coming out at night because the reptiles couldn't see as well at night. So they're kind of not completely like cave fish, but they were pretty much not too reliant on being able to see color because they were mostly coming out at night and they were living underground. So 2 of those genes died. So we know why a gene dies. We know because it's not important, right? Question is, if you look at what happened then to us primates, is we went, because we know we're mammals, so somehow primates acquired an extra copy of the gene for seeing color. We went from 2 to 3. So if there are ways to kill genes, there must be ways to make them or make extra copies. And the main mechanism is through a process called a duplication. Okay? Now, duplication is exactly what it sounds like. You make an extra copy of a gene. An interesting example of that is going back to when we looked at reading a gene. So the gene that we looked at, if you remember, what was the name of the gene that we looked at? Do you remember its name? It was oxytocin. Okay. Now, there is another gene called vasopressin, also known as antidiuretic hormone, which we'll talk about in the next unit. If you look at the amino acid sequences of these 2 proteins, you'll notice that they're both 9 amino acids long. There's the first one, and there's the last one. And 7 of the 9 are the same. Okay? They're very, very similar to each other. And that's because they both descended from a gene called vasotocin. which we find in things like chickens and reptiles. Okay, what happened to vasotocin is it got duplicated, so an extra copy. And in mammals, one of the vasotocins was modified through mutations to become oxytocin, and the other one was modified through mutations to become ADH or vasopressin. And that's why it's called, by the way, notice the name, vasotocin, right? So vasopressin and oxytocin are actually descended from vasotocin through a duplication process. There are many examples in our genome of this. There's one. I'll show you. The other one is for seeing color. And what actually happened is the yellow gene. If I go back to mammals, it says mammals have two copies that work to see color, blue and yellow, okay? The blue copy is actually on chromosome number 4. So this one's actually on chromosome number 4. The yellow copy is actually on the X chromosome, okay? Now, what happened in primates, and by the way, it's not all primates. I think in most primates, I can't remember exactly where it started, but in most primates, the yellow copy got duplicated, all right? And now what happens then is the yellow copy, if you make 2 copies, all right, one through mutation became the gene for seeing red, and the other one was modified to see green. These are the red-green opsin genes that sit on the X chromosome. If you remember from your Mendelian genetics, you might have remembered things like sex-linked inheritance, where males are more likely to have issues seeing red color or green. It's because males only have one copy of the X, right? So if it's defective, then let's say they have a defective X for seeing red, then that individual is going to have a hard time seeing red or things that have red in it. We know this happened because when you look at the gene for the red and green opsins proteins, or the actual just the DNA sequence, they're almost identical, kind of like oxytocin and vasotocin. And that tells us if they're almost identical, that means that they came from an ancestral gene, in this case, the yellow gene, okay? So we talked about different types of mutations. We talked about killing genes, and we've also talked about, right now, making genes. So let me end there. Let me stop the recording, and then if you have any questions, we can